Sanibonani guys, and welcome back. In a previous lesson, you were introduced to the concept of mutations. You learned that mistakes can occur in the DNA code. Mutations occur spontaneously in all living cells. Occasionally, mutations will occur when the individual has been exposed to some environmental toxin or nuclear or X radiation. The individual's DNA will be irreparably damaged. Some mutations have very little effect on the organism. Some may be lethal and cause the death of the organism or some disorder, but other mutations may even help the organism. Two main types of mutations exist: gene mutations, which are also known as point mutations, and chromosome mutations. In a previous lesson, you learned that gene mutations involve the changing of single base pairs. These mutations occur when a copying error happens during DNA replication. Sometimes incorrect bases are substituted or bases are added or deleted from the DNA chain. In this part of the lesson, you will learn about chromosome mutations. Meiosis is a very complicated procedure. It is quite remarkable that it actually goes right most of the time. Sometimes, however, meiosis does become faulty. Abnormal meiosis will result in chromosome mutations. Faults during meiosis can result in a chromosome changing or even a number of chromosomes in a gamete changing. When chiasmata form during prophase 1, a piece of chromosome can break off. This can get lost or reattach itself incorrectly. Quite a few genes will be involved. Depending on the genes that have been lost or incorrectly attached, the mutation can be mild or severe. These are some examples of the chromosomal mutations that can take place. In deletions, one or a few genes are deleted from the chromosome. In insertions and duplications, part of a chromosome is duplicated and then inserted into the chromosome. In inversions, part of the chromosome breaks off and is then reinserted, but back to front or inverted. In translocations, part of one chromosome breaks off and becomes reattached to a completely different chromosome. Another kind of chromosomal mutation is called non-disjunction. During prophase 1, instead of the homologous pairs going to opposite poles, both pairs of one chromosome can move to the same pole. The result is that a gamete can be formed with too many or too few chromosomes in it. One example of mutation brought about by this kind of faulty meiosis is Down syndrome, or trisomy 21. A person with trisomy 21 will have 3 copies of chromosome number 21. This person may have varying degrees of mental retardation, poor muscle tone, tendency to heart conditions, as well as a characteristic physical appearance. There may be small almond-shaped eyes. Ears are situated lower than normal. There is a large flat forehead, flat nose bridge, large tongue, and short stubby fingers and toes. The risk for a woman to have a trisomy 21 child increases with her age. This suggests that meiosis may become more faulty as an organism increases in age. An incorrect number of chromosomes caused by an incomplete set or extra chromosomes is called an aneuploidy. Down syndrome is an example of an aneuploidic disorder. Most cancerous cells carry many extra chromosomes and may even be polyploidic. Polyploidy means there are extra whole sets of chromosomes, not just some extra chromosomes. A cell with 3N, 4N, 5N, or etc. is a polyploidic cell. In a future lesson, you will study a variety of genetic disorders which will result from mutations. That's it for me, guys. I hope you enjoyed the lesson and see you next time.